Hey, this is Jackson Harrell, and I'm going to be presenting on how prostaglandin E2 can act as a modulator of viral infections. So to get started with this, we want to make sure that we understand all the background information going into this, so what the immune system is, how viral infections work. So we're going to start with a quick recap of some important information just at basic level to get us started. So we have the viruses versus the immune system. And viruses, as you probably all know, are small infectious agents that cause disease in all life forms. There are many subcategories of these viruses, with distinctions being made on the number of strands, whether it's single-stranded or double-stranded, and whether or not they are DNA or RNA viruses. Additionally, we want to understand that we have immune systems that protect us from these viruses and other pathogenic conditions. Organisms have evolved these immune systems over millennia, and these immune systems are divided into two types. So we have both these types in us, but we have two types of immune system. We have our adaptive immune system and our innate immune system, each with different key players like lymphocytes, natural killer cells, interferons. And then we also have compounds that aren't immune cells, but they have activity that regulates or modulates our immune responses. And so, just a quick understanding to make is that our adaptive immune system was one that, like the name suggests, we adapted over time. We encounter antigens like different pathogens, things like that, so that our body can recognize them and develop responses. And so, but our innate one is one that we're kind of just born with. It's very complex, especially given all the modulators we have. If you actually look to the right side of this slide, I've inserted a graphic from the paper I'm doing this presentation on, and it kind of outlines how different immune cells, different modulators act together to create a plethora of different immune responses. There's so many different interactions. It looks like it's chaos. It's very convoluted, it looks like, but it's actually very specific, and it allows for extreme variability in our responses, which is part of the reason why our adaptive immune system is so readily able to fight off different infections. So one of these key modulators, though, we want to really focus on, which is a type of prostaglandin. Specifically, as the title of this presentation suggests, it's prostaglandin E2. But first, we need to understand what prostaglandins are, which brings us to our current course material. So prostaglandins, as you probably know from class, they are arachidonic acid-derived lipid molecules, and they are produced by cyclooxygenases and prostaglandin synthases. They are key modulators of our immune system, and they are actually a type of prostanoid, which is in turn a subclass of eicosanoids, something you probably remember from class. There's a slide. The next slide is actually one of our course slides that I'm going to use to kind of draw this back to our current course material. So looking at this, we know that eicosanoids are signaling molecules made by the oxidation of arachidonic acid or other polyunsaturated fats, and they're found in nearly all mammalian cells, which kind of tells us that these have to be really important if they're found in nearly all of our cells. And if you just look at that graph at the bottom I made that on my iPad for y'all. It kind of just helps you visualize. So these are a bunch of big words, so it's kind of easier sometimes to visualize how the classification or the hierarchy of it goes. So prostaglandins are prostanoids alongside thromboxanes and prostacyclins, and prostanoids are a type of eicosanoid. Going to our next slide, we have one from class. This is our slide on the classes of fatty acid derivatives. So we have eicosanoids, which are made of 20 carbons, and they're derived, like I stated previously, from arachidonic acid or other 20 carbon lipids, which is what we meant when we said that they can be made from other polyunsaturated fatty acids. As you remember, we have prostaglandins, we have thromboxanes, but we also have leukotrienes and lipoxins, resolvins, eoxins, all different types of fatty acid derivatives. And you might also remember this next slide from class, which is one that shows how these eicosanoids are created. I have a later slide after this one that goes into further detail on its biosynthesis. But if you look at this, you see that arachidonate, or also known as arachidonic acid, is released from our phospholipid membranes and can then either undergo a cyclic pathway or a linear pathway. And so we can make prostaglandins, thromboxanes, lipoxins, resolvins, leukotrienes, or eoxins. As you can see on this graphic right here on the right, like I said, this slide is taken from one of Dr. Vance's slides, and it just kind of helps you visualize the great variability that can be attained from taking this arachidonate from the phospholipid membrane. So let's get specifically, though, into prostaglandin E2. We haven't talked about it yet, and that's what this entire presentation is about. So prostaglandin E2 is one of the most studied prostaglandins out there. This is because it's produced by fibroblasts, macrophages, malignant cells, and it regulates a lot of bodily processes via the PGE2 receptors. That's the prostaglandin E2 receptors. And these four receptors are EP1, EP2, EP3, and EP4. So the prostaglandin E2 it serves as a very important regulatory role on the immune system in response to various viral infections. And so therefore it shows a lot of potential as a potential therapy for the treatment of viral infections. But in addition to just this viral infection response, PGE2 also plays a big role in other responses in our body and different just necessary bodily processes, like regulating blood pressure, gastrointestinal integrity, fertility, and inflammation responses. Those are all very key processes to our continued survival, so obviously this prostaglandin E2 is very important in the human body. Now, we need to understand how does prostaglandin E2 get made. So looking at the biosynthesis of it, it's first started, as that previous slide showed, by the liberation of arachidonic acid from membrane phospholipids via phospholipase A enzymes. That's our PLA2 enzymes. And there's three subclasses of this enzyme, but there's one we want to focus on, which is CPLA2 alpha. And it performs this reaction of liberating arachidonic acid from the membrane as its primary function, and it's found in almost every tissue in our body. So the importance of that fact is that if this thing serves with its only function is to remove arachidonic acid from the membrane for the synthesis of prostaglandins, and it's found in almost every tissue in your body, it means it has a vast variability in its importance and its roles in our bodily processes. So keep that in mind when you look at this. Hey, this is a pretty important guy in our bodily functions. And as I previously stated, we have those cyclooxygenases and those prostaglandin E2 synthases that are very important for turning the arachidonic acid into prostaglandin E2. And so cyclooxygenases are what we'll start with first, and they are membrane-bound heme-containing glycoproteins with two primary functions: the addition of 15 hydroperoxy group to arachidonic acid to make prostaglandin G2, and the reduction of nascent hydroperoxy groups to form prostaglandin H2. So we get prostaglandin G2 and H2, but that's not what we want. We want E2. Well, these two prostaglandins, G2 and H2, are substrates that are used in the synthesis of PGE2 from PGE2 synthases. So we use cyclooxygenase to get our arachidonic acid, which is released by that phospholipase A. And once the cyclooxygenase has it, it can go ahead and add that 15 hydroperoxy group to the acid, and it can form prostaglandin H2, or it can do the reduction of the nascent hydroxy peroxy group to make PG H2. We have different mechanisms that can make precursors or substrates for PGE2, which is a more stable form of our prostaglandin. So I know that's kind of convoluted. I'll say it again. We have phospholipase releases arachidonic acid, cyclooxygenase makes prostaglandin G2 and H2, and then PGE2 synthase turns that G2 and H2 version of prostaglandin into the most stable PGE2. So it's kind of a little bit of a long process, but you see how they act one after the other, and each one is a kind of a downstream flow to make PGE2. And an important note to make on this process is that our cyclooxygenases have two isoforms. There's isoform 1 and there's isoform 2. Isoform 1 has the same function as isoform 2, but isoform 1 uses arachidonic acid derived from dietary intake, whereas isoform 2 will use endogenous arachidonic acid, so stuff we already have. And then another important statement to make sure we all understand is that PGE2 is degraded very rapidly in vivo, meaning in the body, via 15-hydroxyprostaglandin dehydrogenase. And if you look at the right side of the screen, you can see this whole process of releasing arachidonic acid from the phospholipid membrane, and you can see the pathway it takes and the various different pathways it can take to make different compounds. So this is a very intricate pathway that's really cool to look at, a little bit confusing to try to understand fully without understanding all the background information, which is why I am presenting all this background information. Now, we have our PGE2 though now. We've made it from our PGE2 or PGH2 via PGE synthase. So now we want to figure out how do we use this PGE2, because if it just sits in the cell, it's useless. It's made intracellularly. We need to get it outside to act upon other bodies. So, as I just stated, it's produced intracellularly and requires a method of excretion in order to act upon extracellular bodies. Initial theories postulated that PGs exit the cell via passive diffusion, but the kinetics behind it did not back up this hypothesis. It showed that that couldn't be true. And we later found out that prostaglandin transporters and the multi-drug resistant protein 4, PGT and MRP4 respectively, were found to import and export PGs. So both of those proteins are integral proteins, meaning they pass completely across the membrane, which makes sense if it's going to take something inside and move it outside or vice versa. And it's important to note that PGTs are only found in prostanoid-producing cells, but that MRP4 is found in all cells, which, of course, it is a multi-drug resistant protein. So now we know how it leaves the cell. How does it act upon another cell? How does it induce that response? Which is what brings us to the PGE2 receptors and the signaling. So as I previously stated, PGE receptors are EP1, EP2, EP3, and EP4. And there's a lot of information to go into this. There's a lot of variability in the pathways they use, the actions they perform. There's extreme variability, and these different variants of the receptors allow for adaptable patterns and responses for various cell types at different stages of immunity. So you can encounter all sorts of different antigens, different pathogens in your life, and that's why it's so important that we have a vast array of options and combinations that we can use. And just a list of types of receptors that we can look at. We have the EP3 and EP4 have the highest affinity for prostaglandin E2, and so thus they can be active at way lower PGE2 concentrations compared to EP1 and EP2. EP2 and EP4 will mediate the anti-inflammatory response and suppressive PGE2 activity via G-coupled receptors, which are in turn mediated by CAMP, PKA, and the Krebs pathway, as you probably all remember from different information we've learned in Biochem 1 and Biochem 2. And then we have that EP1 and EP3 are not dependent on the G-coupled receptors. Instead, EP3 has a cyclic AMP inhibiting function, and EP1 signals via calcium ion release. And then lastly, we have that EP1 and EP2 are involved in the degranulation of mast cells, which in turn induces vascular permeability and local edema. And it also induces congestion thanks to those two symptoms. If you look at the right, we have the different receptors, EP1, 3, 2, and 4. And we also have adenylate cyclase and the calcium channel PGE2 receptors, transporters. And these just show the different functions and how PGE2 can move through these pathways. As you know, it's biochemistry. Biochemical pathways can be very complicated. They're heavily regulated. And if you want to pause this just to look at all the different effects of the different receptors, it's very interesting to look at. You can see the stress responses, pain transmission, joint inflammation, cell migration. It's really cool just to look at. And so I'd highly encourage looking through this article if you want to look at the charts alone. They are heavily detailed. I've used them throughout this presentation. And now that we know how these receptors work, how it's transported, we want to understand what role does PGE2 have on inflammation and immunity. I've been telling y'all this the whole time, but what roles does it actually have? Well, all classic signs of inflammation are due to prostaglandin E2. It causes redness, edema, augmentation of arterial dilation, microvascular permeability to increase blood flow. It does a lot. And all the pain associated even with inflammation is induced by this action on the peripheral nervous system, peripheral sensory neurons, and on the central sites within the central nervous system. Prostaglandin E2 also binds to those EP receptors to regulate the immune function of macrophages, dendritic cells, lymphocytes, and it allows for pro and anti-inflammatory responses. So it can kill inflammation or it can cause inflammation. And so essentially that binding of prostaglandin E2 to any of the EP receptors can induce a differentiation of immune cells to perform certain immune responses, including antigen recognition, phagocytosis, and immune cell activation or suppression. It does it all. And additionally, given its role in regulating the immune system, research suggests that this prostaglandin E2 might even play some role in autoimmune diseases and disorders. And if you look to the right there, it actually, this chart that's also brought from the paper I'm reporting on, shows the specific effects of PGE2 on different types of immune cells. So if you want to pause it and read those, you can see the various effects that it does have on them. Now we have PGE2's role specifically with viruses. So it doesn't just cause things to happen to the immune system, it also can do things to the viruses. So in addition to working with the body to maximize immune responses, it can have a positive or negative effect on viruses. So, you might have been thinking this whole time, oh gosh, PGE2 does all this work for us on our immune system. It's so great. Well, it also has an effect on viruses. It can help them out. The variation in its roles on viral prolificacy is largely dependent on the class of virus and the host factors. So if you look at that chart on the right, which again is taken from the paper I'm reporting on, you can see that based off the class of virus, there's different research that's been done on specific types of those classes of viruses that can show how it affects its the expression of the virus, its reproduction, everything. And another factor that complicates our understanding of the role of PGE2 on viruses is comorbidities, because sometimes when there's multiple viruses or disorders going on, it can have altered effects. And so if you want to pause this just to look at it, that chart is actually really cool to look at how prostaglandin E2 can affect different viruses, such as human immunodeficiency virus, herpes virus, Epstein-Barr virus. It has a significant effect on them. And then moving on now, if we know that it has an effect on viruses, sometimes positive or negative, then that means that we could probably use it as a therapeutic agent. So if we have prostaglandin E2, we know that it has a stimulatory and inhibitory effect on viral life cycles and the immune system. And cyclooxygenase isoforms are responsible for the downstream production of it. So we could target PGE2 production. We could target any factor that contributes to it. So we had those cyclooxygenases, we had those prostaglandin synthases, but unfortunately it makes it, because it has dual stimulatory and inhibitory effects, it makes it very, not difficult, but at least you have to be very meticulous with what you're doing, because you might cut off one pathway that's causing a disorder or disease, but by doing so you might also activate some other pathway. So there's a lot of research being done to try to understand how prostaglandin E2 could be used as a therapy or as a treatment for different viruses. And so with that note, I'm going to go ahead and pose some questions to you guys just to theorize on. You should be able to answer these based off the information given in this PowerPoint. But if you'd like to understand them further, if you have any trouble with it, you can ask me if you'd like to. You can email me, or you can refer to the article in question. So our first question would be, given that prostaglandin E2 can assist viruses, a way to stop this effect would be to stop its production. Based off of this presentation, limiting ingestion of what compound could reduce PGE activity? And if you did reduce said compound, which cyclooxygenase would see a reduction in activity? So I just kind of told you a big hint by saying that cyclooxygenase would see a reduction in activity, so maybe refer to that slide. And then question 2 being, what effect could an increase in concentration of calcium ions have on mast cells, and what physical symptoms would be observed from this interaction? Additionally, which receptor is involved in this process? So there's a lot to, that sounds like a lot to answer, but just if you understand one part of it, it's kind of a chain reaction. So just go through and review this presentation, read the article if you like, but this presentation and the abstract should be sufficient. And I look forward to seeing y'all's responses. Thanks so much, and y'all have a great day.